MSCs, they solve a very different problem. They solve this problem very differently. So we are not making the MSCs recognize the tumor antigen per se. We are not asking the MSCs to become immune cells. We are using the MSCs as delivery vehicles. And the primary job of the MSC is not necessarily to kill the tumor, but its job is get to the tumor and bring things with it. So MSCs, they naturally traffic towards inflammatory and damaged tissue. So they can tolerate hypoxic conditions pretty well, and they localize within the tumor microenvironment.
What if the cell wandering towards sites of damage in your body could be re-engineered into a precision cancer therapy? Mesenchymal stem cells have intrigued the field for decades, but turning their natural tropism into a reliable therapeutic vehicle has proven hard. Today I'm joined by Jun Yung Woo, co-founder of AGEM Bio, to explore how his team is engineering MSCs into a fundamentally new kind of treatment. Let's dive in. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brühlmann, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Welcome, Yung, to the Smart Biotech Scientist. It's good to have you on today.
Hi, David. Thank you so much for having me. It's definitely an honor to speak with you.
Yung, to start us out, share something that you believe about bioprocess development that most people disagree with.
I think one thing that I strongly believe, and I know that not everyone agrees, is that bioprocess development scales too early. So in biotechnology, we are naturally drawn to numbers. How many cells can we produce? What's the yield? What's the cost per dose? And can we move from T-flask to stirred tank bioreactors and so on and so forth? So those are very important questions, but I think they are often asked before we've fully understood the biology. At the end of the day, we are not manufacturing monoclonal antibodies. We are actually manufacturing live cells. So this process isn't just producing the product. The process is really shaping the product. So give you MSCs, for example. You can put them on microcarriers. You can generate billions of cells. That's relatively straightforward. But have we asked a simple question? What happens to these cells after 10, 15, 20 population doublings? Have we looked into how the expansion really changes their secretome, their migratory properties, their immunomodulatory functions, or even their susceptibility to engineering in our case? So I've seen certain situations where teams become extremely good at manufacturing the cells themselves, but haven't fully established whether these cells that they're manufacturing are still biologically effective. So for me, the order should actually be reversed. You should first understand the cell, understand what really drives the potency, then understand what causes the loss of function, and then build a manufacturing process around preserving those attributes. Otherwise, there's a risk that you may actually be scaling wrong biology very effectively.
Yeah, you're making an excellent point, and there is a big distinction between biologics and cell and gene therapy. We're going to dive further into the complexity of particular cell therapy and stem cells. Before we do that, draw us into your journey, Yung. What first pulled you towards cell therapy, and what were some key moments along your career path?
AGEM Bio wasn't created because we just wanted to create a company. It was really a combination of nearly 2 decades of scientific work. So before I started my PhD, our lab actually has already developed a highly efficient non-viral engineering platform. At that time, the challenge we were trying to solve was really fairly straightforward. How can we modify difficult primary cells efficiently without using viral vectors? So that technology worked really well for MSCs. We became the starting point of my PhD. Initially, we weren't focused on building the company, or we weren't even focused on developing the therapeutic product in the first place. So we were more fascinated with a fundamental question of what actually happens to the cell after you engineer it. Well, a lot of people focus on transfection efficiency. They ask questions like, did the gene get in? And things like this. But we became increasingly interested in the engineering process. So what were the stress pathways that were activated? How did those stress responses actually influence the cell's function, the persistence, the potency, and even the manufacturability of these cells? Over the course of my PhD, we spent a lot of time actually dissecting this cell biology of engineered MSCs. We discovered that many of the challenges that people attributed to poor transfection efficiency were actually downstream consequences of cellular stress, responses, intracellular trafficking, bottlenecks, and so on and so forth. So at the same time, we also realized that if we engineered MSCs, if these engineered MSCs were ever going to be real medicines, right, the biology itself was not enough. We needed the manufacturing, we needed the preservation, we needed the scalability, and so on and so forth. Alongside this biology, we actually built many different technologies for scale-up, for cryopreservation, formulation, for quality control, and so on and so forth. What surprised me really was the breadth there was in such a platform. So the more we learned about MSC biology, the more these possibilities actually emerged. And we started exploring different therapeutic payloads, different mechanisms of action, and different disease indications. And gradually it became clearer and clearer that it wasn't just a transfection technology anymore. So this was becoming more of a therapeutic platform. That was actually the turning point. And we began generating compelling efficacy data. First, we did some preclinical studies, and then later on, we actually progressed to doing some of the studies in companion animals with naturally occurring cancers. And then at that point, we actually faced a very practical reality, academic funding. So academic funding is excellent for generating these sort of discoveries, but it is actually not designed to really bring these therapies into patients. So if we wanted to move beyond the publications, we wanted to move beyond just getting papers and so on and so forth, we really needed to actually develop a medication. We needed a different vehicle. And this vehicle is actually ultimately became AGEM Bio. So the company was created to translate everything that we had learned from the cell biology, from the engineering, the manufacturing, and the various preclinical observations into a new generation of MSC therapeutics. So in many ways, we are still following the same scientific question that really started this journey. How can we turn MSCs into programmable therapeutic vehicles by preserving everything that makes MSCs biologically unique?
Yeah, that's exciting. And Jung, tell us what makes MSCs or mesenchymal stem cells compelling as a therapeutic vehicle, because they've been around for quite a while now, and I'm sure many people are familiar with them, but why do we use them?
MSCs are actually one of the most fascinating cell types in regenerative medicine because they really challenge our traditional understanding of how cell therapies work. So when people hear about stem cells, they hear the word stem cell, they often imagine the cell replacing the damaged tissue, right? But actually, that is not what MSCs do. So over the past 2 decades, we have learned a lot about how MSCs work, and a lot of their therapeutic function actually comes from the signals that they secrete rather than the ability for them to differentiate. So MSCs are essentially biological sensors, biological responders. They detect inflammation, they detect injury, they detect hypoxia, they detect tissue stress. And in response to that, they actually secrete a complex mixture of cytokines, chemokines, growth factors, extracellular vesicles to help modulate the local disease environment. What's exciting about all of this is that many chronic diseases are actually fundamentally diseases of a dysfunctional microenvironment. Inflammation that has become chronic, repair mechanisms that fail, immune responses that become dysregulated. So rather than targeting a single pathway, MSCs actually influence many of these pathways simultaneously. So that is why the field has explored MSCs over such a broad range of indications. And we have seen some approved products. And we've seen encouraging clinical data in graft versus host disease, inflammatory bowel disease, osteoarthritis, diabetic wounds, critical limb ischemia, so on and so forth. But the challenge, however, is actually it has always been consistency. So how can you make these therapies consistent? And that has ultimately led us to what we have been developing, which is MSC 2.0.
Okay. Before we talk about the specifics of your platform, can you tell how MSCs are different from other cell therapies? Because we know there are CAR Ts, there are NK cells, there's a lot more TCR and so on. What are the differences?
Probably one of the most important questions in cell therapy today, because MSCs, they offer something unique. So MSCs are different from immune cell therapies. CAR T therapy has been really transformative. So the results have been very good in certain blood cancers. But the biology of blood cancers, they are fundamentally different from the biology of solid tumors. So there have been many hematological malignancies, and they are relatively homogeneous. And a large fraction of the tumor cells, they actually express the same antigen. But solid tumors, they are completely different. They're heterogeneous, they're hypoxic, they're immunosuppressive, and they are spatially very complex. So using something like an immune cell is very difficult to target such heterogeneous population. So that's actually what ultimately attracted us to the use of MSCs. MSCs, they solve a very different problem. They solve this problem very differently. So we are not making the MSCs recognize the tumor antigen per se. We are not asking the MSCs to become immune cells. We are using the MSCs as delivery vehicles. And the primary job of the MSC is not necessarily to kill the tumor, but its job is get to the tumor and bring things with it. So MSCs, they naturally traffic towards inflammatory and damaged tissue. So they can tolerate hypoxic conditions pretty well, and they localize within the tumor microenvironment. So by engineering therapeutic payloads for the MSCs to carry, we can potentially achieve a very high localized delivery while minimizing systemic toxicity. That's a very different philosophy from immune cell therapy.
How did you develop your platform? What does it look like today? And how does your platform work?
The platform that we have developed essentially is we have a non-viral modification platform. Initially, it was developed for the modification of neurons, actually, then for primary cells. And we realized that MSCs in particular, we are able to transfect MSCs really well. This actually led us to delve further into it. The field, as we know, primarily they modify MSCs, they use viruses. And viruses, they have evolved for over millions of years to solve a delivery problem. Viral systems are very efficient. But what we realized in the course of this research is that delivery actually wasn't the bottleneck. The bottleneck was actually the trafficking within the cells itself. Even if the materials, as you know, materials, nucleic acids, if you deliver it to the cells, a lot of times they are just sequestered within the endosome and ultimately they are degraded. So if this material, we are able to rescue it from the endosome and help it to navigate into the nucleus to allow for expression, you really get a very robust transfection or modification. So that's how we actually develop the system. We have a fusogenic lipid, which promotes the endosomal escape. And we have a HDAC inhibitor, which stabilizes the microtubule transport system in the cell. So this allows for intracellular trafficking. And together, these two interventions greatly help to increase or enhance the transfection efficiency that we see in our MSCs.
And how does then the platform work, or your engineered MSCs? What kind of payloads do they carry, and how eventually do they fight the different diseases you were trying to treat?
For oncology specifically, we designed the MSCs to carry two payloads. The first is cytosine deaminase, and the second is actually a cytokine called interferon beta. So this cytokine, interferon beta, fires up the C-gasting immune pathway. So we are simultaneously delivering these two payloads in a single cell. So why did we choose these two is because we designed it to work a single unit, a single system. So we call the product AGEM 102, and it's designed to target the fundamental mechanisms in cancer biology. So at a conceptual level, cytosine deaminase generates tumor stress. Interferon beta converts that stress into an immune signal. So that is the overall framework. cytosine deaminase converts 5-FC, which is the prodrug, into 5-fluorouracil locally within the tumor microenvironment. This creates a very highly localized chemotherapy wherever the MSCs accumulate. So 5-FU would then disrupt DNA synthesis, RNA synthesis, and induce tumor cell damage. But what interested us was not just this cytotoxic effect, but it's actually a lot of the downstream biology. So when tumor cells experience DNA damage, DNA fragments accumulate and they form abnormal in the intracellular compartment. And this activates a lot of the innate immune sensing pathways. One of them is a very prominent one, is actually the C-gas-STING pathway. Activation of this pathway can promote type 1 interferon responses and actually recruit immune cells to stimulate anti-tumor immunity. So there was a study done by Prof. Jun Woo's lab in Yale University that actually demonstrated this very elegantly. And the challenge is that many aggressive cancers, including the cancer that we are trying to target, which is glioblastomas, they have evolved a lot of mechanisms that actually suppress this C-gas-STING pathway. Some of them silence STING, some of them have epigenetic silencing of a range of the different components of these pathways. Others even have deletions, chromosomal deletions, whereby the whole interferon cluster is deleted. So in those situations, tumor damage alone may actually not be sufficient. So we asked ourselves this question. So what if tumor damage and we simultaneously supply the missing immune signal? So what if we could do that? That is why we actually ultimately designed AGEM 102 to deliver these two payloads simultaneously. So one arm, the chemo arm, damages the tumor, whereas the other arm, the immune arm, which is the interferon beta, creates noise that allows the immune system to then recognize these damages.
You have now a powerful MSC that is engineered carrying these payloads. How do you make sure that on one hand, the cells find the tumor and not some healthy cells? And on the other hand, as MSCs naturally navigate toward inflammation and tissue damage, that they don't end up at the wrong space?
This is actually one of the first questions that our regulators asked us, and rightly so. So whenever we deliver such a potent therapeutic payload, the question is not so much if it works. The question is more of whether you can control it and control where it works. So what's important to understand is that AGEM 102 isn't a constitutively active cytotoxic therapy. The chemotherapy itself requires the administration of the 5-FC. Without the 5-FC or without the prodrug, cytosine deaminase actually does nothing. So it doesn't do anything. So that immediately gives us a level of control, first level of control. The second layer comes with the localization. So we are deliberately administering the MSCs as close to the disease site as possible. So the goal here is to concentrate the therapeutic activity where the residual tumor cells are most likely to be found. So that's the second layer of control. The third layer of control really comes from tumor biology itself. Rapidly proliferating cancer cells, they're generally much more vulnerable to 5-FU compared to normal tissues. So they have higher rates of proliferation, they have greater genomic instability, they are often defective in their DNA damage response pathways. What we are really doing is we are stacking these multiple layers of selectivity, the local delivery, the prodrug activation, and tumor susceptibility. So we understand that no therapy is perfectly selective, but together these factors actually create a therapeutic window that we've been able to demonstrate in a lot of our preclinical studies. And we hope that this is ultimately what we can demonstrate in our phase 1 study as well.
And how long do these engineered cells actually stay at the tumor site? Because as they're in that environment, for instance, the T cells are shut down or some other immunosuppression is going on, and your payload is exactly designed to overcome that. But obviously a critical factor, your cells need to be there sufficiently long time in order to reverse or even eventually kill all the cancer cells. How does that work?
We've done multiple studies in animals, in some dogs and cats, and in preclinical mice studies as well. We find that for a majority of the preclinical mouse studies, when you inject the MSCs, they typically do not persist for more than 14 to 21 days. So that's about 2 to 3 weeks. And after that period, they get washed out. Another thing that really is part of the therapy design is that the MSCs themselves are also sensitive to prodrug. So if you feed the 5-FC, the MSCs convert it to the cytosine deaminase that is in the MSCs, convert it to 5-FU, and the MSC itself also dies from that 5-FU. They really is sort of like a kill switch that you do not need to worry so much that you will leave a footprint. So that's how we really designed this study as well.
And tell us a bit more about your clinical studies with the dogs and cats. How did you design the study? Are there some interesting stories or things you have observed?
For the dogs and cat, there will probably be one particular case that I'll remember for the rest of my career. It was around 2019. This was pre-pandemic. There was a dog that actually presented in the vet clinic with a recurrent perianal adenoma. So this is very aggressive tumor around anal region that is very common in smaller breed dog. This perianal adenoma in this particular patient actually has already recurred multiple times, and the patient has already undergone multiple surgeries, but each time the tumor recurred. So by the time we became involved, the surgical options were actually becoming increasingly difficult. So I remember I personally delivered the cells to the vet clinic, and the vet performed the intratumoral injection of these stem cells, and they scheduled a follow-up a week later. So when I came back the following week, I was actually genuinely surprised. We have seen it in mice models, but seeing it in a real patient is actually really quite remarkable. I really noticed that, there was a visible regression in the size of the tumor. So it wasn't just a subtle regression. It actually shrunk sufficiently such that now surgery became feasible. I mean, we know that scientifically this is one case. It doesn't prove much, but we have to be careful not to overinterpret these sort of results. But from an emotional standpoint, that was probably the first moment that we realized that, wow, this technology actually does have a future beyond the laboratory. And it really transformed the project from an academic exercise into something that was more tangible. It really has a potential to help change lives. And that's a very, very wonderful feeling.
Absolutely. And speaking of having a future, once you treat, right at the moment, these animal patients, is there a durable protection or will the cancer come back?
This is one of the most fascinating questions we have encountered. So the short answer is that we do not know definitively, but we have certainly made observations that make us really curious. So several animals that we have treated experienced durable responses long after the engineered cells were gone. So even after we've done the dose for months, they still remain cancer-free. So in some cases, we do not see recurrence for over 4 to 5 years. We've also observed what appear to be abscopal effect. So for example, when we treat one site and then another site responds. So we had this one case of a patient with oral melanoma, and the oral melanoma had actually metastasized into the lung. So due to difficulties delivering into the mouth directly, we actually treated the lungs first, and that was via IV administration of the cells. And to our surprise, not only did the lungs improve, but the oral lesion actually shrunk. So those observations really raised the possibility that, anti-tumor immunity has been generated. But the challenge is that these are compassionate veterinary cases, and we don't have the luxury of doing serial biopsies, of doing T-cell analysis at a larger scale, and immune monitoring, and so on and so forth. So what we can say is that these observations are consistent with the possibility of immune memory, but we cannot say that we have proven it entirely. That is exactly one of the questions that we would love to answer in a lot of our future studies.
The promise of MSCs has always run ahead of the engineering needed to deliver it. And what Jun Yung Woo has shared shows just how much that gap is starting to close. There is a great deal more to unpack in part 2. If this conversation gave you something to think about, please leave a review on Apple Podcasts or your favorite platform. Thank you so much for tuning in, and I'll see you next time. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.